Hi, my name is Dr.
Gaukhar Baidildinova.
This video is a part of my lectures on evidence-based medicine and epidemiology. Uncontrolled studies. Uncontrolled trials lack a control group, meaning all participants receive the intervention. This design is commonly used in phase 1 trials to assess the pharmacokinetic properties of a new drug. However, uncontrolled trials tend to overestimate the effectiveness of an intervention compared to controlled trials, leading to inflated expectations. Due to the higher risk of systematic error, their results are generally considered less reliable than randomized controlled trials. Another major concern is their use in conditions that may self-resolve, which can further exaggerate treatment effects. For example, in immunotherapy for warts, uncontrolled studies should be avoided. Since warts can disappear on their own, using this design may falsely attribute their resolution to the treatment, compromising the validity of the results. Controlled studies. Controlled studies are clinical trials that include a control group, allowing researchers to compare the effects of an intervention against a baseline, e.g., a placebo, standard treatment, or no treatment. Why are controlled studies important? They help eliminate bias, improve reliability, provide stronger evidence of cause and effect relationships, and ensure that treatment effects are not due to chance, placebo, or self-recovery. There are several types of comparative trials, each designed to evaluate different aspects of a drug's effectiveness. Superiority trials assess whether the investigational drug is more effective than the control. Equivalence trials confirm that the drug's effect is neither better nor worse than the control within a predefined margin. Non-inferiority trials determine whether the investigational drug is at least as effective as the control. Dose-response trials examine the relationship between dose and effect, helping to identify optimal dosages, including both the initial and maximum tolerated doses. I also have 2 paid courses on Udemy. The Critical Assessment of Clinical Studies teaches how to read and critically evaluate clinical studies. It's a very great course. The same as my second course, Scientific Writing, where I tell all the nuances of how to write a scientific article or dissertation. Links are in the description box. There are 5 types of controls: historical, lack of treatment; placebo, active or synonymous positive control; and dose effect. Historical controls, external and non-simultaneous. Control sources are external to the present study and were treated at an earlier time, earlier therapeutic gold standard, or in other conditions. Historical controls have the advantage of studying rare conditions where creating a sample is difficult. A disadvantage is that randomization or blinding is not possible in this design. Medical practices evolve over time, decreasing groups' comparability. Another disadvantage of this design is the difference between baseline characteristics of subjects in the experimental group compared to the historical group. Parallel, no treatment control. In this design, no intervention will be given to the control group. Study endpoints should be objective. Disadvantages of this design include potential observer bias and difficulties with blinding. Active or positive control. In many cases, it may be unethical to give a placebo to a person with a disease. In this case, a previously approved treatment with known efficacy is administered to a control group. This design can be used to demonstrate equivalence, not less efficacy and superiority. The Declaration of Helsinki mandates the use of standard treatments as controls. This design is most ethical when approved drugs are available for the disease being studied. Dose effect. Concurrent control by comparing doses. It is used in dose-response trials. This design uses different doses or regimens of the same treatment as the active and control groups. The aim is to establish a relationship between the dose and the efficacy, safety of the intervention. This design may include active and placebo groups in addition to groups with different doses. It may not be effective if the therapeutic range of the drug is unknown.